Okay, we'll be starting in a couple of minutes. Uh, Good morning, everybody. We'll be starting with the third session today. Let's see what do we have. So initially, we will be talking about PCR. What is a PCR? What happens in a PCR?
Sir, amplification of the DNA molecules.
Right, right. Amplification of DNA is what happens. So this is essentially the exactly how PCR works. And this is only the cycle that has been given. Before this, there is an initial denaturation phase. And after this, there is a final extension step. So essentially, there are primers which are forward and reverse primers, which bind to the DNA and amplify the target gene or target, the DNA that you want to amplify. So if you want to observe the real time how amplification looks like, you perform a qPCR or a real time PCR, which has uh, cyber green dye. It's a dye based chemistry. There is probe based chemistry also. So do you know anything about that? Which kind of dye is used?
Ethidium bromide.
Ethidium bromide is used for qPCR, or is it used for agarose gel electrophoresis?
That is used for agarose gel. This one, wait.
Cyber green. Cyber green dye is used in dye-based chemistry. There are other dyes also. And you know anything about probes, probe-based chemistry?
Some molecular marker type of molecules,
We
marker type
use
of molecules
satellite.
which may be radio labeled.
Have you heard about these? TACMAN molecular
So what I have
beacons,
written is
TACMAN probe or molecular beacons.
no sir.
So they are very similar to how your primer is designed. These kind of probes are designed so that they specifically bind to your DNA of interest, and they will give you fluorescence. So in a dye-based chemistry, cyber green dye, it binds to a double-stranded DNA. Hence, whenever double-stranded DNA is produced as a result of a polymerase chain reaction, there will be fluorescence, and we can read out this fluorescence, and we get how much amplification is done at the end of each step. We can find it out. Is that clear?
Yes, sir.
Okay. Then there is something called a melt curve. So, do you know what is a melt curve? This is exactly the first diagram shows the melt curve.
That temperature versus fluorescence intensity. Uh,
What is here and what is here?
Basically, the temperature in which the DNA
Temperature is
denaturation.
increasing. Fine. What else?
And as the temperature increases, the denaturation, the DNA starts to denature. So at a particular point of temperature, 50% of it is denatured.
That
That
is
is
the
the
melting
melting temperature.
temperature.
Melting temperature.
So this is the dsDNA. I mean double-stranded DNA. Here it is single-stranded DNA, right?
Yes, sir.
So in between you, somewhere around here, this will be the TM value for the, I think this is the wild type. Then this is the mutated, let's say, phenotype that you're observing. Then there will be, sorry, reduction in melting temperature. So you can essentially find out that with the difference of melting temperature, there is a change in the genomic content of the DNA. So what kind of change would you expect to come up on a melting temperature? What would you, anybody knows about it?
The melting temperature is the temperature at which 50% of DSDNA gets denatured.
That is fine. That is fine. What I'm trying to say is what kind of change in the DNA would have an effect on its melting temperature,
The base composition,
right?
the salt concentration, presence of intercalating dyes or intercalating fluorescent dyes. These are some of the factors.
Essentially, the GC content.
The GC
If there is
content.
a change in GC content, if GC content is increased, then what will happen? The melting temperature will increase or decrease?
It will increase.
Right. Fine. That is how we find out whether there is a change. So once a PCR product is formed, we then run it in an agarose gel electrophoresis. I assume all of you understand what is an agarose gel electrophoresis. There is a ladder, and corresponding to the ladder, we have our test samples by which we actually calculate what is the size of our amplicon. Right. That is how we do. Then this is a process of cloning. When a lot of people were asking about cloning. So there is a plasmid vector. Then there is a target gene that you have amplified using PCR, and then you have run the gel, and then you have done gel extraction, and you get this target gene. Now you digest the plasmid and the target gene with the same enzyme, and then you join them together using ligase, and this reaction is called ligation reaction. So why do we have to digest the plasmid and the target gene?
Because plasmid is plasmid to host as a vector, and and we will insert that gene of interest or target gene into that plasmid, which actually belongs to some other organism maybe. So to transfer that gene, we need to cut the gene from the genome of that organism, and then we have to inject it here. But if the plasmid is not broken, then how can we join it, because it is covalently closed circular. So therefore, we need to digest both the target gene as well as the plasmid with the same enzyme, so that uh, they have complementary sticky ends, which and hence can join with the help of DNA ligase.
Complementary sticky ends is the correct answer here. So it will increase your chances of clone production of the clone. I mean, this is the clone we are talking about. Then this clone is either transformed into a bacterial system. It could be transduced using some bacteriophage as well. That is called transduction. And if you insert this clone in an eukaryotic cell, then it's transfection. Is that clear?
Yes, sir.
Okay, now this gene that you have, Riddhi, you have a question?
Yes, sir. Is it possible for a target gene to not have the complementary sequence at all to the plasmid?
target gene never has a complementary sequence. Essentially, when you are amplifying using a PCR, when you are amplifying your target gene, you insert the restriction site manually.
Okay, so it's not that something's already present and you're just removing the things around it. It's something you're adding.
Yes, yes, yes. So if this is the gene, let's say it starts, let's say from G, T, C, A, G, something like that. And you know, EcoR1's, so when you will design the primer, you will design it according to this, right?
Yeah.
So before this, I will add, let's say, EcoR1 site, GATTC. That is for EcoR1, right?
Okay.
And I will add 3 random nucleotides. This is done because, you know, so that the enzyme can actually sit and work and recognize, I mean, the site. So you are manually inserting the restriction site while you are preparing the insert DNA for cloning. So, and this same site will be present in the plasmid also.
Okay.
Is that clear, or is there some
So I
confusion
think
left?
what we've learned so far is that they recognize the sequence that are already present and cut around it. So how do you add it manually? Like how do you program that?
Program, see, when you are, you know, the restriction site. This is the restriction site of EcoR1. You know that?
Okay.
So when I think this is the restriction site, am I saying something wrong? I don't know. As far as I remember, this was the
Yes, sir.
huh?
Double A haven? Is it
Sir,
double A?
একটা A, B, C, D.
Okay, it
Double
could
A?
be double A. Yeah, I'm sorry.
GATTC.
GAATTC. Sorry. Okay. So GAATTC, this is the I mean, EcoR1 recognition site. So when you will synthesize your primers, when you will give it for that synthesis part is a chemical synthesis. That is a separate thing. But when you design your primer, you send it for synthesis, you will have the restriction sites added. So whenever your primer will, you know, wait, in this step, when your primer is sitting like 5' to 3', in the 5' end, there will be an overhang which will have the restriction site. Here also, in the 5', there will be overhang which will have a restriction site. So your amplicons will have, let's say this is the amplicon, and before that there will be the restriction site.
Okay.
Understood?
Yes, sir.
So, okay, I think that is clear. And then we will talk about Western blotting, Northern blotting, Southern blotting. So essentially what happens, some of you said SGS page is used for proteins, to separate proteins. So we will separate them and then we will transfer them into a membrane. That happens again through an like a process similar to electrophoresis. There are charges. So the protein is then transferred to the membrane. Now, this membrane could be nylon membrane or PVDF membrane. These two membranes are usually used for Western blotting. Sorry, it is nitrocellulose membrane or PVDF, not nylon. Nylon is used for Northern blotting. Now, then we will block this. That protein will stay there. Then we will wash. Then we will add primary antibody. So the primary antibody will essentially bind to a specific protein that you are looking for. We will go through certain experiments after this. We will understand what is the role of a primary antibody, and then we wash it again, and then we add a secondary antibody which will bind to the primary antibody and will also help us visualize for detection purpose. And this is how a band of Western blot is observed. Is that clear?
Sir, can you please repeat this overview?
So first, you take the protein, isolate proteins, run the gel, Fine. Then you transfer it into a membrane. These proteins are then transferred into a membrane, following which you will block the membrane so that the proteins they stay where they are. Then you will wash it. After every step there is washing. I'm not saying washing continuously. So you will add the primary antibody. So primary antibody will only bind to the specific protein of your interest. So why do we do this? Say if you have taken a cellular lysate. Cellular lysate has a lot of proteins. Everything will come up in the blot. But you want to only find, let's say, a particular protein. Let's say catalase. So you will use an anti-catalase antibody that will only bind to catalase. So you can only locate, you only want to visualize the catalase. Understood? Then there will be a
Yes, sir.
secondary antibody which will bind to the primary antibody, and it will give you uh, it will help you visualize.
Sir, if the primary antibody is enough for recognizing the protein of interest, so what is the utility of the secondary antibody binding?
Secondary antibody has the label, no, for detection. Primary antibody does not have that.
Okay, sir.
Understood. Okay. So similarly, the workflow is also given on the right hand side. So you essentially load the protein sample. This is how a page apparatus looks like. Then you transfer the protein from the gel to membranes. You probe with antibodies. So there is a primary antibody which is target specific, and then there is a conjugation secondary antibody. So conjugation with secondary antibody. Then you capture the image in a gel dock and you get your blot. So we'll see the blots later. Then there is southern blotting where there is agarose gel electrophoresis, following which again there is a transfer to membrane, nitrocellulose membrane. Then what you do, you add radioactive probes. So this is the process of hybridization where your target, let's say target DNA. Based on that, you will have the probe. So the probe will have a radioactive label. So then you will expose it to an X-ray film, and the radioactive labels they will develop. They will be marked as your blot in the blot. Clear?
Yes, sir.
Okay. So similarly, we have northern blotting also. It is done for RNA. So western blotting for protein, southern blotting for DNA, and northern blotting for RNA. So here you do an electrophoresis for RNA, then you transfer it into the membrane, and then you visualize it. The same process for this also. Today we will be discussing about two papers only. So this is the first one about the pathology in MNSOD mutated mice. So MNSOD is SOD2, mitochondrial SOD.
Yes, sir.
We have seen it similar things what has happened in case of cardiac myopathy earlier in the previous paper, in the previous session. So today we will see what happens when there is MNSOD uh, mutant. So essentially what happens, what they have removed, again there are 5 exons. They have removed the first and second exon, if you see in the mutant. Is that clear?
Yes, sir.
Okay. So they have removed it, and because of which the wild type. Okay, and they have inserted this PGK HPRT, this sequence. I mean, this is a particular insert that they have inserted. It also has the same role as that of a selectable marker that has increased this length of the entire mutant. So now the mutant is 8 kb, wild type is 5 kb. So if you see only the wild type, this part, so it has only 5 kb bands. But if you see any homozygous, let's say mutant, this one, it only has 8 kb bands. Is that clear? Why
Yes,
that
sir.
is happening? Okay. So in mice, you see similar pathology. Which one is mutant? Which one is normal?
Smaller one is mutant.
Right. Fine. Then you see the SOD2, activity. SOD2 activity when seen through a Western blot. What is observed in the plot B? Anybody?
For homozygous dominant, the we have
Wild
a
type.
corresponding
Wild
wild
type
type
or mutant?
homozygous. Wild type.
Okay.
So for a wild type homozygous dominant, we are having a band for SOD2 and as well as 28S and 18S of rRNA. And but for the homozygous recessive, which is uh, or which might be the case for the normal only, we will not have that particular SOD2 gene. So since we are mutating the gene, it is similar to that.
So you see a band here.
Yes, sir.
Is there any band here?
No, there is no
So
band.
it's in so in a wild type, there is SOD2 expression. In the mutant, there is no SOD2 expression. I can say that.
Yes, sir.
Then why is 18S and 28S done? 28S and 18S, they have the same result in all the three cases. Why are we doing this?
Because it might be the fact that 28S and 18S are they are ribosomal RNA, so they are not being changed, right? So they will always appear, and a non-functional protein is formed from the mutant, which actually does not have the function of SOD2. So we are getting a corresponding bands for 28S and 18S, but not for SOD2 in the mutant strain.
So what if you do not get the band for 28S and 18S also in the mutant?
Wait, sir.
What?
Wait, sir. I'm thinking.
That means you have done something wrong with the experiment. So do you know the concept of control? You take a control, like apart from your protein of
Yes, sir.
interest, you
Yes,
will
sir.
have to check a control protein that you know will express in all the cases. So let's say if you cannot develop a blot for the control, that means you have done something wrong with Western blotting. So the result which you're inferring with your protein of interest is probably not correct, right?
Yes,
And
sir.
understood? Okay, that's it. So when we see the weight of the mice compared with the age they have lived. So if you see the wild type, there were 9 mice and the heterozygous mutant. They all appeared to have similar pathology, similar weight, and they live for similar time. I mean, they live for the entire duration of the experiment when it was conducted. But the homozygous mutant, SOD2 mutant, they have a lower weight and they die on the 13th day. So it's like if you remember 10 days after birth, they die. So similar to that. So is that clear?
Yes, sir.
Okay. Then coming to if you see the hematopoietic cells, this is in case of the mutant. This is the case of wild type. So you will see that there are much less cells here compared to this. It has reduced hepatic glycogen. They are, not evenly spread like in D. It is evenly spread in the panel of D. But here it is not evenly spread. There is kind of an accumulation. Even there is lipid accumulation. So, glycogen, I mean, How is glycogen converted to glucose? What is the process? You have studied it in biochemistry.
Glycogenolysis.
So glycogenolysis
Glycogenolysis.
and lipid metabolism, these things are also affected due to the change in MNSOD. Okay, so this is something which we observe. So here also SOD2 gene was, knocked out, but it was a different approach, right, than the previous one. There they used a neomycin resistant gene. Here they have just increased the length of the mutant sequence. So it appears that SOD2 is a tumor suppressor gene. So if we knock down the expression of SOD2, there is malignant tendencies which develop. So this is not with reference to this paper. There are certain other articles which talk about it. So there is impaired expression. It's impaired expression. So if you reduce the expression of SOD2, then it causes development of ovarian cancers and type 1 diabetes, things like that. So it not only affects cancer progression, but also the metabolic the condition of the patient. So if we overexpress SOD2, it has been observed experimentally that if you overexpressed it, then tumor formation is reduced. Clear? Okay. What were the key findings? SOD2 were knocked out, and these mice were severely anemic. So as you can see in this slide also, the mutated ones, they had a lot less hematopoietic cells compared to the wild type. So they had less RBCs. So they were anemic. You know what is anemia? Then there were degeneration of neurons in basal ganglia and brainstem. It is not shown as a diagram, but it was observed. There were progressive motor disturbances in the mice which had mutated SOD2. Dilated heart was observed in the transverse section. So what is this pathology called?
Dilated cardiomyopathy.
Right. Very good. So electron microscopy showed extensive areas of cell death. The mitochondria were swollen and it caused, the cell to undergo apoptosis. The vertebral bone marrow cells of the knockout mice, they showed reduced hematopoietic cells, so which explains the severe anemia. We have already seen it. There is enhanced lipid deposition in the liver of the mice. There is an increased damage in the cells and tissues which have requirement for high oxidative metabolism. SOD activity, while it was reduced, there was an increase in SOD1 and SOD3 activities. That is CUZ and SOD. Why does this happen?
To compensate for the loss of MNSOD.
That is correct. And then there is demodulation or reduction in activity of aconitis and succinate dehydrogenase. Why does this happen? Anybody remember the last session?
The coenzyme Q gets changed.
Is it by
No, wait.
citric acid cycle?
They are present in citric acid cycle, yes.
So what's the question?
There is a marked demodulation or reduction in the activity of aconitis and succinate dehydrogenase due to the knockout of MNSOD or mitochondrial SOD superoxide dismutase.
So what is demodulation? What's demodulation?
Reduction in activity. What will happen if mitochondrial SOD is not present?
The enzymes, I mean, the DNA will be damaged, for which there will be less production of enzymes and less activity. Maybe
You
I'm not sure.
remember
I don't know.
this?
Yes. Acha, you will go.
Or that
Study you will go on to
superactive oxide
right, right.
voltage.
Sorry.
Yes, sir. Yes.
So it has
Four,
four
four.
Fe4S clusters. Aconitis and succinate dehydrogenase has four Fe4S clusters. We have discussed this in the previous class.
Yes, sir.
So when there is a higher amount of peroxide radical, which is not converted to hydrogen peroxide anymore because MnSO3 is absent, so it is attacking this uh, 4Fe4S clusters. You remember?
Yes, sir. And it
Okay.
is getting converted into 3Fe4S, I guess.
Yes.
Yes.
So this conformation is non-active.
Yes,
I
sir.
mean, the enzyme doesn't work in this case. So is it clear to everybody?
Yes, sir.
Okay. So then again, growth and development of the knockout mice are normal in the uterus of mother. Why? We have discussed this. When the mice is
No
growing,
metabolic activity for the mice inside.
yeah,
It's
low
very low.
metabolic activity, right?
Low
Yes,
metabolic activity.
that is right. So the oxidative damage is very less. So then this is the next paper that we have, familial cardiomyopathy with cataract and lactic acidosis. This is actually a case study, fine. In pediatric research, it was published. Now, if you see, cardiomyopathy is an autosomal recessive disorder. Its onset of symptoms can be during the birth, it could be during the childhood or early adulthood. So what are the manifestations in case of cataracts and cardiomyopathy? You know what is a cataract?
Yes, sir.
Yes, sir.
So cataract, it could be congenital, that is coming from your previous generation to you to the next generation. Mitochondrial myopathy could be there. Myopathy essentially is like there is a defect in the muscles essentially. That is what is myopathy. So in cardiomyopathy, there is hypertrophic cardiomyopathy. So in this case, the heart muscles, they are thickened. Instead of getting thin, they will become thick. And then there is a variable onset of this disease. It can come, as I said, it can happen during the birth, it could happen during the childhood or early adulthood. So it's variable and it's progressive. So once it starts upon the onset, the disease condition will increase. I mean, it will be progressive in the sense is what it means is with age, the disease condition will become more severe. Then there will be constitutive lactic acidemia with slight muscular exercise. You know what this is?
Very quick accumulation of lactic acid with even slight exercise or
So
muscle
usually
activity.
when you have a good workout session, you
Cramp,
have the sorry.
cramps.
Cramps, yeah, because of the accumulation of lactic acid. So here we have talked about metabolic acidosis. So here similar thing happens, and lactic acid is accumulated much faster in this case. So in hypertrophic cardiomyopathy, there is isolated hypertrophic cardiomyopathy by autosomal dominant genes, which are responsible for beta myosin. Some maternally inherited forms of cardiomyopathy happens due to mutation in mitochondrial DNA and tRNAs for glycine and leucine. So if there is hypertrophic cardiomyopathy with lactic acidemia, there is an impaired mitochondrial respiratory chain. So if there is an impaired mitochondrial respiratory chain, there will be an impaired oxidative phosphorylation. Essentially, whatever happens in mitochondria will not work properly. So pyruvate will be fed into a lactic acid cycle instead of citric acid cycle. Is that clear?
Yes, sir.
Okay.
Sir, one more thing that how is lactic acid is getting accumulated so fast? We know the process by which lactic acid might get accumulated due to vigorous physical exercise, but how come
What
like
happens when you
small
have rigorous, when you do a rigorous exercises?
lactic acid accumulates because the cells, the muscular cells, they switch to anaerobic respiration. So, uh, but in this case, what means with very slight muscular exercise, how is this happening?
Why does it change to anaerobic respiration?
Because if it means if it has to get energy in a very short amount of time, and
So the
for mitochondrial.
oxygen available is not sufficient for oxidative phosphorylation to
Yes,
happen,
sir.
right? So the
Yes,
ATP
sir.
produced is not enough.
Not
So
enough.
we have to go through the lactic acid cycle so that we can
Yes, sir.
produce enough ATP. Now, if the mitochondria is dysfunctional, now you have enough oxygen for sure,
Okay,
but
okay.
the number of
Understood.
mitochondria is
Understood.
less.
Understood.
Understood?
Understood. Understood.
So this is the situation. I mean, this is what is this called? This kind of a uh,
AT3 chart.
yes. Have you studied about it yet in genetics?
Last day in lab we did it.
Lab mein. Acha, there is an experiment. I remember. Okay. So what happens? This family is a consanguineous family. What it means that they have, like marital relationships between their cousins, their siblings, essentially. So as what you can see in for the fourth generation, very specifically. So the people in the fifth generation, they died. They had cardiomyopathy and cataracts. So autopsy was done on patients V3 and V7, and for the rest of them, the autopsy was denied. Okay, so usually this kind of situation used to arise in royal families, where they used to think to for the continuity of pure blood, they need to have royal associations. So earlier these kind of diseases used to come up very frequently. Nowadays it is less, but still there. Usually these kind of diseases are recessive in nature, they do not happen. But if you have this kind of a situation when there is a consanguineous family, then there is a higher chance of development of this kind of disease. Can anybody tell me why in terms of genetics?
Sir, can you repeat the question?
A recessive disorder is more likely to be present, I mean, to appear if there is consanguineous relationship. I mean, uh, if you have, let's say, siblings are getting married together, having kids, then there's a higher chance that there will be a, development of a recessive disorder.
So because they already have that recessive gene and they are being the carrier of it, so there are 50% chance that they will transfer it to the next generation, and it has a higher probability that the since both of them have the recessive allele, the next generation will get the recessive allele more likely.
Yeah, correct. That is correct. That is correct. So you're right. So in the patients V3 and V7, we observed few things. First of all, we saw citrate synthase activity. So citrate synthase is nothing but the control enzyme here, just similar to what we have seen in the Western blot slides also, that we require a control so that we know. Essentially, when you're extracting a protein out of a cell, uh, if you just measure its activity as such, you will not know it is because you have extracted more amount of protein, whether your extraction efficiency is better, or whether it is because actually the protein is produced more in a cell. So for that, we use some other protein that will be expressed in either of the cases, and you normalize the expression with that protein. So they have done the same thing in the cardiac muscles. You see, there is not much difference between the skeleton, I mean, between the patient and control. But if you see in the skeletal muscle, there is not much difference. But in case of cardiac muscles, you will observe that the ratio of C1 and C3, C1 and C3 is complex 1 and complex 3 of the mitochondria is much less in patients compared to that of the control. Same we observe for C2 and C3. Even if you compare individually, then there is a marked reduction of these activities compared to the control. Control is the normal healthy patient. Is that clear?
Yes, sir.
Okay, now we see the lactate to lactate to pyruvate ratio. Okay, so the lactate to pyruvate ratio increases in patients, essentially V3, V7, whatever it is increasing. Why do you think that happens?
Because the lactic acid accumulates very fast as compared
So,
to amount of pyruvate. So therefore, the
so,
lactic acid amount increases much more.
so whatever pyruvate is produced in the sense in the end of glycolysis is fed into the lactate system. The lactic acid is produced. Similarly, if you observe these are essentially cytochrome activities in C2 and C3 and C4. So there are differences. There is a reduction in activity in C1 and C3, C2 and C3. There is increase in activity. So something like that. There are some changes, not very important. And the ATP production using different substrate, when we give different substrates, then we observe that the ATP production is essentially it is increasing in patient V7 and patient V3 also. It's kind of, I mean, it's not increasing or decreasing. Actually, these values are changing, but it is not significant. So you know what, when is a value significant and when is it not? Have you heard about statistics? I mean, studied it yet?
Yes, sir. A bit.
So when there will be a lot of changes in our biological systems, but you have to do statistical tests to realize whether this change is due to an effect of some change in the pathology or some disease, or it is due to chance alone, because there is a good amount of standard deviation between any two biological species. So there was not a significant difference in this case. So that is what we observe. And then this is the cardiomyopathy. I mean, in the autopsy report, they did this Western blot. So they used an anti-complex 1 antibody. So this is the primary antibody. You remember the Western blot slide?
Yes, sir.
This is the primary antibody. So what will it determine? What will it, I mean, bind against?
It will bind to the complex one of
Ah,
the
right.
mitochondrial respirators.
That is true. That is correct. So we observe here that I mean, just to tell you that complex 1 has a 75 kilodalton, 42, 30, 24, and 20. These are the proteins, subunits essentially, which are present in complex 1. So if you see the diseased condition and the healthy patient, there is not much difference. So there is protein production in a similar rate. So complex 1, in that case, we observe. In this case, you see this is again a Western blot where you are seeing against anti-MNSOD and anti-CUZNSOD. So in case of MNSOD, there is an overexpression. Can you see here you cannot see anything, but here it has increased.
Yes, sir.
24 kilodalton. And so around 24 kilodalton is the weight of the MNSOD protein, and around 33 kilodalton is the weight of the CUZN-SOT protein. It's actually little more than that, 34 or 35. But anyway, so this is the control, a normal healthy patient. This is in case of V7 and V3. They all have CUZN-SOD expression. Not much change. It is similar. I mean, you might say it is reduced or something like that, but it is not significant enough. I mean, these kind of assays are not very good for quantitative analysis anyway. So then we observe the MNSOD mRNA transcripts. What would be the technique which could be used here?
Northern blot.
Right, right. That is correct. So it again has 4 kilo base pair and 1 kilo base pair. What is it called? Uh, I mean, there are two bands essentially corresponding to MNSOD, and there also we observed that in patient V7 specifically there is an higher uh, MNSOD expression. I mean, transcript levels compared to control, and V3 as well. So V3 also has a higher level compared to control. And then this is the southern blot. So essentially what happens, this is the control. And this is for the skeletal muscles, this is for the cardiac muscles, right? You digest the DNA fragment or the DNA with the same enzyme. Let's say you're digesting here. I think I don't know which enzyme they used. I mean, which restriction site they used. I'll have to look into it, but you will digest it using the same restriction enzyme. And in this case, there is only one band in the control situation. In skeletal muscles also there is one band, but in cardiac muscles there are multiple bands. It appears like a smear. So what might this signify? Anybody? I mean, what does this mean? Okay, let's try to visualize this. So this is a normal DNA, right? You have multiple copies of it, and these are, the two restriction sites. Let's say R1, R1. These are the two restriction sites which are present. So let's say this is the length of this is, let's say, 16 kb. Fine. Is that clear to everybody? No responses. How would I know?
Sir, can you repeat once more?
You did not understand this part entirely?
No, sir. I actually my network connection went out actually.
Okay, so let's try to visualize what is happening here. This is a DNA fragment which has the same restriction site and at the end of it, and the length is 16 kb. Is it okay?
Yes, sir.
Till here, okay. So you observe that you will get a 16 kb band in the gel. That is how it should be.
Yes, sir.
But in some case, you're observing that there is a 16 kb band which is, predominant. Then there is a 14 kb band. Then there is a 12 kb band, 10 kb band, 9 kb band. So what does this mean?
It means that some amount of DNA is being damaged and broken apart.
What kind of damage?
Deletion.
Yes, deletion, right. So that is exactly what happens here also. So essentially, there is a deletion of certain. There is a deletion in the DNA of these, cardiac muscles. Which DNA they have seen? Just let me have a look. I'll have to see. Just give me a moment. Just a minute. Must have written it here. Okay, this was mitochondrial DNA. So mitochondrial DNA in the cardiac muscles, they had suffered deletions. So if there is a deletion in the mitochondrial DNA, that is essentially genomic instability. Now coming to the MNSOD activities, MNSOD activities in the control were like this. It has increased in case of patients. MNSOD activity is increased. We have seen the same thing in the Western blot, in the Northern blot as well. And this assay actually gives you the rate of superoxide production. So the rate of superoxide production was higher in that of control patients. In patients, it was lower because of higher MNSOD production. So that is what happens. So what we see that we see what are the changes in the mitochondria due to hypertrophic myopathy. That is what we observe in this paper. And then there is more number of mitochondrial, the numbers of mitochondria increase in the heart muscles of the patient who have this disease, right? Um, so that is something we observed in the southern blot. We observed that there is DNA deletion in the mitochondria in the patients, and further investigations they revealed that they have the higher MNSOD levels compared to normal fibroblasts. So I mean in the normal patients. So any questions?
Sir, what is this mitochondrial etiology? What is etiology?
Yeah, the condition of mitochondria, what happens in mitochondria. So usually
Oh, okay.
people have studied this with respect to, cardiomyopathy, with respect to what happens in the heart, in the heart muscles, and what happens everywhere else. But they did not study it with reference to what happens in mitochondria. So this study actually told us that if you remember the previous slides where we said कि mitochondrial dysfunction leads to cardiovascular diseases.
Yes,
So
sir.
this is one of the examples. So this is a case study. Okay, this is just a report where they found these changes. So when they saw MNSOD levels were changed, and mitochondrial DNA had suffered damage. So they understood that if you usually what we have seen, we have knocked out MNSOD and we have seen that some diseases are forming. But in this case, there was a disease and we observed that, okay, the mitochondria was not functioning properly.
Okay, sir.
Anything else?
Sir, I have one basic question, you might say. That is, I have actually forgot the link between the mitochondrial dysfunction and which leads to cardiac myopathy. So that part can you explain once more?
Where did we have it? I think in the second or
I guess it was in the second day PPT. I
let me
couldn't remember
try.
exactly.
I also think actually I did not explain anything with pertinent to that. But if you
Like
remember,
how is the dilated condition of the heart is arising due to the mutated MNSOD maybe. So which leads to
okay.
mitochondrial dysfunction.
Here, if you remember, CVD, cardiovascular diseases are coming up, chronic kidney diseases, all are happening due to mitochondrial dysfunction.
Yes, sir.
This is one slide we had, and the later we discussed about the paper about where did it go? Yeah, this one. So here, what did we do? We inactivated SOD2
Yes, sir.
and we knock out of SOD2, and then we observed the heart, and there was dilated cardiomyopathy that was observed in this case. Even in case of hypertrophic cardiomyopathy, we observe that there is an, if there is a defect in SOD2, it will lead to that as well. So A lot of diseases are coming up. I mean, what do you need to know here?
Uh, I just needed to know whether it is known that what is the basic mechanism by which mitochondrial dysfunction is causing uh, dilated cardiomyopathy. If it
No,
is known, if
I
it has
mean,
been tracked.
in this paper, it was not discussed. Essentially, we are just, in a classical geneticist approach, when we see when a gene might have an effect on something, sort of disease, we knock out that gene and we see if that disease is happening. And if we overexpress that gene, if we will see if that disease is reducing or something like that. I mean, you understand? So this is what we have done in this. So regarding the mechanism exactly, maybe there might be some other papers who might have tried to understand. So essentially, in these papers also we see that more number of mitochondria are coming in. Which they are trying to compensate for the less number of mitochondria which are present already. Then there is apoptosis also in the cells. So these kind of things are happening. But exact mechanism I cannot tell you. I mean, I don't know.
Okay, sir. I actually asked this because, like we know that if that SOD2 gene, the gene for SOD2, if it gets mutated, it leads to mitochondrial dysfunction, and we are seeing in the rats that in general, their body size is decreasing, but how it is causing dilation of the heart actually. That's why I was a bit
It
confused.
is also causing dilation in some cases. It's also causing a thickening of the heart muscles in some cases. How is everything
Yes.
happening? We do not know, but we will definitely see I mean, that is a part of the next session, that when we overexpress this SOD2 in Drosophila, the lifespan of Drosophila increases. So it's a teaser for your next session. So I will just stop the recording here if it is okay with all of you.
Yes, sir.
So we increase what? That
It's
increases
Life,
the lifespan.